Hello and welcome to Easy Learning with Dr. Salman Khan. Next, morphology of Plasmodium. It shows the following forms: Sporozoite, Hypnozoite, Merozoite, Trophozoite, Schizont, and Gametocytes. The features in each of the forms differs in all the 4 species of Plasmodium, with few species showing similar features. The features explained further are for an easy and comparative understanding of the forms in all species. Morphology of each of the 4 Plasmodium species will be dealt in separate videos. First, about the Sporozoite. The name is derived from the Greek word sporos, which means seed. It is a minute, thread-like curved organism, tapering at both the ends. It measures 9 to 12 micrometers in length, has a central elongated nucleus, and does not contain any pigment as seen under a light microscope. This is the diagram of the sporozoite with all the structures it possesses: a nucleus, endoplasmic reticulum, cytoplasm, convoluted tubules, Golgi apparatus, mitochondrion, pellets, rhoptres, microtubules of pellicle, and the apical cap. Electron microscope studies of sporozoite reveals the following features. There is a thick double membrane, the outer corrugated and the inner stout. At the anterior end, there is a cup-like structure lying in an apical depression. 12 hollow peripheral fibrils are attached to the cup. These fibrils are found to be contractile or tensile in function. These have a locomotory function. A long bulbous paired organelle presumed to secrete a proteolytic enzyme to facilitate sporozoite's penetration of cells in both hosts. The nucleus lies in a well-marked pit called the micropyle. Inside the cytoplasm are numerous mitochondria which provide a source of energy. This is the sporozoite as seen under electron microscope. This diagram shows the anterior end of the sporozoite of Plasmodium falciparum, which consists of the following structures: apical cup, concentric rings, peripheral fibril, pellicle, mitochondria, and a paired organelle. These are the sporozoites when stained and seen under a microscope. Next, Hypnozoite. This name is derived from the Greek word hypnos, which means sleep. In Plasmodium vivax and Plasmodium ovale, hepatic form known as hypnozoites persist and remain active in hepatocytes for considerable time, which may be from months to years, before they grow and undergo pre-erythrocytic schizogony with the liberation of merozoites in bloodstream, causing lapse of the infection. Size: they are about 4 mm in diameter and are uninucleate. In latent stage, in Plasmodium vivax and Plasmodium ovale, 2 kinds of sporozoites are seen, some of which multiply inside hepatic cells to form schizonts, and other persist and remain dormant in the resting phase. In relapse, the resting forms are called hypnozoites. From time to time, some are activated to become schizonts and release merozoites, which go on infecting RBCs, producing clinical relapse. These are the hypnozoites when seen under a microscope. Next, Merozoite. The name is derived from the Greek word meros, which means a part, and zoon, which refers to animal. This is the next developmental stage of sporozoite. This takes place within the liver cells. Merozoites are pear-shaped bodies about 1.5 micrometers in length, possessing an apical complex called the rhoptre. The merozoites infect the erythrocytes by a process of invagination. They attach to the erythrocytes by their apex, and then the merozoites lie within an intra-erythrocytic parasitophorous vacuole formed by red cell membrane by a process of invagination. These are the merozoites when seen under a microscope, when stained. Next, Trophozoite. They are the cytoplasmic mass, blue in color. They are seen in the RBCs. They contain the malarial pigment called as hemozoin. The trophozoites are of 2 types. One is the ring form, also called as early trophozoite, and the band form called as the late trophozoite. Ring form or the early trophozoite. In the erythrocyte, the merozoite loses its internal organelles and appears as a rounded body having a vacuole in the center with the cytoplasm pushed to the periphery and the nucleus at one pole. These young parasites are therefore called the ring forms or young trophozoites. Since the cytoplasm is thinner at one side and thicker on the other, it is named as ring form. This is seen in Plasmodium ovale, Plasmodium vivax, and Plasmodium falciparum. The parasite feeds on the hemoglobin of the erythrocyte. It does not metabolize hemoglobin completely and therefore leaves behind a hematin globin pigment called the Malaria pigment or hemozoin pigment as a residue. This is the ring form when seen under a microscope. then band form or the late Trophozoite. As the ring form develops, it enlarges in size, becoming irregular in shape and shows amoeboid motility. This is called the amoeboid form or late Trophozoite form. Since the cytoplasm is uniformly thick, it is called as band form. This form is seen in Plasmodium malariae. This is the late Trophozoite or the band form when seen under a microscope. Next, Schizont. This is the product of the process of schizogony. It is of two types, immature Schizont and mature Schizont. When the amoeboid form reaches a certain stage of development, its nucleus starts dividing by mitosis, followed by a division of cytoplasm to become mature Schizonts or meronts. A mature Schizont contains 8 to 32 number of Merozoites and Hypnozoite. The mature Schizont bursts, releasing the Merozoites into the circulation. Immature Schizont, these are oval in shape and the size is larger than an RBC. The nucleus is divided into 2 to 4 parts and the cytoplasm is undivided. This is the immature Schizont when seen under a microscope. Then mature Schizont, this also is oval in shape. The nucleus is divided into 12 to 24 parts and the cytoplasm is also divided. Each nucleus is surrounded by a portion of cytoplasm. This form contains Merozoites. This is the mature Schizont when seen under a microscope. This is containing 6 to 12 Merozoites, on an average 8. The infected RBC is smaller than an uninfected RBC. Next, Gametocytes. These are of two types, male also called the microgametocyte and the female called as the macrogametocyte. After a few erythrocytic cycles, some of the Merozoites that infect RBCs do not proceed to become Trophozoites or Schizonts, but instead develop into sexually differentiated forms, the Gametocytes. They grow in size till they almost fill the RBC, but the nucleus remains undivided. Development of Gametocytes generally takes place within the internal organs, and only the mature forms appear in circulation. The mature Gametocytes are round or oval in shape, except in Plasmodium falciparum, in which they are crescent shaped. Fusion occurs leading to formation of a zygote, which later forms an O-kinnate, forming an oocyst, and finally formation of sporozoites. These are the features of male and female gametocyte. Male gametocyte is round in shape, crescent in Plasmodium falciparum. Nucleus is large, loose, and in the center, malarial pigments are diffuse and prominent. After staining, the cytoplasm appears pale blue or pink, and the nucleus appears pale. In female gametocyte, the shape is oval, the nucleus is small, compact, and not in the center. Malarial pigments are concentrated. After staining, the cytoplasm appears dark blue and the nucleus appears deep red. In all species, the female gametocyte is larger. Male gametocytes have a large nucleus that takes up more than half of the parasite, while female gametocytes have a much smaller nucleus. Female gametocytes are generally more numerous than the male. This is the diagram showing formation of microgamete, which involves a process called as exflagellation, and macrogamete. Then the appearance of malaria pigments in different species. In Plasmodium vivax, they appear as numerous fine golden brown dust-like particles. In Plasmodium falciparum, they appear as few 1 to 3 solid blocks of black pigment. In Plasmodium malariae, they appear as numerous coarse dark brown particles, and in Plasmodium ovale, they appear as numerous blackish brown particles. This is the male gametocyte and the female gametocyte when seen under a microscope after staining. These are the malaria parasites under Giemsa stain and a magnification of 2000x, showing erythrocytic stages of the 4 species of Plasmodium. We can see the Trophozoites, late and early stages, then Schizonts, mature and early, and Gametocytes, male and female. This is about the morphology of Plasmodium species. If you wish to see more of such videos, do comment below. And don't forget to like, share and subscribe to my channel. Also, press the bell icon for early notification of my new videos.